Hello everyone, welcome back. So this is week 7 of the NPTEL course Fundamentals of Protein Chemistry, and in today's class we'll discuss about enzyme kinetics and inhibition. Okay, so we'll go through a few topics which are very important for all of your exams, because today we are going to talk about enzyme inhibition, enzyme kinetics, the different models, the different plots. We are going to talk about different types of enzyme reactions, for example ping pong, bye-bye reaction, etc. Okay, and then we will solve questions related to all of these different topics. Okay, so let's begin with today's class. Right, so we'll get right into the Michaelis-Menten equation and how it describes enzyme kinetics. Okay, so I have a slide right after this to tell you how important this is, because a lot of different drugs that we use in our everyday life, these work on the principle of enzyme kinetics. These drugs are designed for very common diseases. For example, the ones that you hear about every day, cancer, and then like different liver diseases and other diseases that you hear about every single day. So drugs designed based on these enzyme inhibition model target all of these different diseases. Okay, so the Michaelis-Menten equation describes how reaction velocity varies with substrate concentration. You know the equation is V naught is equal to Vmax into S divided by Km plus substrate concentration, and V naught is the initial reaction velocity and Vmax is the maximal velocity of the reaction. Km, you know, is the Michaelis constant and its value is K minus 1 plus K2 divided by K1. So where is all this K minus 1 and K2 and K1 coming from? These are the reaction constants of the enzyme substrate product reaction. Okay, and this is the graph for the Michaelis-Menten curve. All right, so now you see, we'll move on to the next slide. So as I was talking about these different drugs which are very commonly used, so you might know the names of these drugs already. So all these drugs work upon enzyme substrate inhibition concept. That is why you understand how important this topic is for our everyday life, right? So for example, aspirin is an important painkiller, right? So what is it doing? It is targeting this particular enzyme, and you can read about this in general. So there are drugs like aspirin, there is drug like statin. So statin is helping with blood pressure control. Okay, and then there is ACE inhibitors. Everybody knows about this because of the coronavirus event recently, right? So all of these different drugs are there which target very common diseases. Okay, now we will move on to Michaelis-Menten equation and the entire concept regarding that. Okay, so now you know that the equation is E plus S gives you ES. So the forward reaction constant is K1. For the backward reaction, the reaction constant is K minus 1, and ES is formed, and then from the enzyme substrate complex, the enzyme plus product gets formed, and this reaction in the forward direction is given by reaction constant K2. Okay, and you saw in the previous equation that Michaelis-Menten Michaelis constant depends upon these reaction rates K1, K2 and K minus 1. Okay, now Michaelis-Menten equation holds true only when we have some assumptions. So these are very important. Okay, so what are these assumptions? Firstly, assumption 1 is that there is no product present at the start of the kinetic analysis. So at the start of the reaction, there is no product, of course. Therefore, as long as we monitor the initial reaction rates, we can ignore the reverse reaction, that is enzyme plus product going to ES. That is why we don't consider this K minus 2, right? So we are assuming that it is starting from scratch. There is no product before the reaction starts. The next assumption is that during the reaction, an equilibrium condition is established for the binding and dissociation of the enzyme and substrate. So this is called Briggs-Haldane assumption. Okay, and thus the rate of formation of the ES complex is equal to the rate of dissociation plus breakdown. Okay, this is the second assumption. So this equilibrium that you're seeing in this reaction this is what is being talked about. Okay, so the rate of dissociation plus breakdown is equal to the rate of formation of the ES complex. Okay. Now there are more assumptions. Okay, so the next assumption is assumption number 3. That tells you that the concentration of the substrate is much more than that of the enzyme. Okay, the enzyme is a catalyst. So catalyst means it is not exactly participating in the reaction. It is just helping with the reaction, and then it is there after the reaction unchanged. Okay, so it is not destroyed and it can be recycled. So only small amount of enzyme is sufficient for a reaction to happen. But the substrate has to be much more than that of the enzyme concentration. Right. So the amount of the substrate bound to the enzyme at any given moment is small compared to the amount of free substrate. That is also an assumption. Then it follows that the enzyme substrate complex is therefore much less than the total enzyme concentration during the course of the analysis. Note, this assumption requires that the reaction is monitored for a very short period, because over time, of course, the amount of substrate will eventually decrease, right? And then this will not hold true. So that is not much substrate is consumed, and the concentration of the free substrate does not effectively change. Okay, that is also an assumption, because your equation depends upon the concentration of the substrate, right? Now, assumption number 4, that is only the initial velocity of the reaction is measured and concentration of the product is 0 initially, and the concentration of the substrate is much more than the initial concentration of the substrate. Okay, now assumption number 5. So there are way too many assumptions already, right? So assumption number 5 is that the amount of total enzyme that is there, it is either present as enzyme substrate complex, or it is present as free enzyme. So E total is equal to E and ES. Okay, the rest of the enzyme is not lost anywhere. It is either this or that. Okay, now we move on to the next slide. So now, based on our understanding of all of these different assumptions and the Michaelis-Menten equation, we come to the conclusion that Michaelis-Menten constant is equal to K minus 1, which is the rate of backward reaction from ES to E plus S, plus K2. K2 is ES to E plus P formation, that forward reaction, and divided by K1. K1 is the forward reaction for ES formation. Okay, and the unit of Km is concentration. So it can be moles, right? Moles per liter or molar. And K1 is rate constant of the formation of the enzyme substrate complex. K minus 1 is the rate constant for the ES complex to form the enzyme and substrate. It's a backward reaction. And K2 is the formation of the EP complex, that is enzyme product complex, from the enzyme substrate complex. So that is once again forward reaction. This one's backward reaction is ignored based on our assumptions. Okay. Now, characteristics of Km. This is also very important. See, Km is the Michaelis constant. It is characteristic of an enzyme and its particular substrate, and it reflects on the affinity of the enzyme for that substrate. Why? Because Km is numerically equal to the substrate concentration at which the reaction velocity is half of the maximum reaction velocity. Okay, that is why it is directly dependent upon the concentration of the substrate. Okay, Km does not vary with the enzyme concentration. It only depends upon the concentration of the substrate. And a small Km. So small Km means what? That even if small amount of substrate has been used, the reaction is reaching half of its maximal velocity. So that means numerically low Km reflects very high affinity of the enzyme for the substrate, because a low concentration of the substrate is sufficient for half saturation of the enzyme. Okay, that is to reach half of its maximal velocity. That means, of course, if you take the exact opposite case, which is large Km, that means a lot of substrate is required for the enzyme to reach half of its maximal velocity. That means its affinity for the substrate is relatively low. Correct. Now we will talk a little more about Vmax. Now this might sound a little boring to you, but this is necessary for your question answers. Okay, that is why it's important that you pay attention. Okay, so the maximal possible velocity or Vmax occurs when all the enzyme molecules are bound with the substrate. So Es is equal to E total. So there is no free enzyme. Okay. And Vmax is equal to K2 into E total. So remember all of these equations because only one value will be given to you and they will ask you to find out all the other values. Okay. So you need to know all these different equations which you can substitute and find out the different values. Okay. So V is equal to Vmax into S divided by Km plus X. This is your normal Michaelis-Menten equation. This is the mathematical representation that is used to model your experimental kinetic data, right? And you also know that Km is equal to K minus 1 plus K2 divided by K1, which is the rate of breakdown of ES divided by the rate of formation of ES, enzyme substrate complex, right? Km is similar but not exactly equal to dissociation constant for the ES complex, right? So dissociation constant also means K off by K on. That means the rate at which two molecules are dissociating divided by the rate at which they're associating, right? So then technically it should be equal, but it is not. Why? Because if K1 is much greater than K2, that means the forward reaction for formation of the enzyme substrate complex is much more than the backward reaction of dissociation of the enzyme substrate complex, then Km is much more than Kd. Okay, because the rate of dissociation is much less compared to the rate of formation. And see, due to this similarity for the expression for Kd, a low value of Km is interpreted as high affinity of the enzyme for the substrate. So now you realize, right? So if it is low value, that means more of ES is getting formed and less of ES is breaking down. And a large value of Km is interpreted as weak affinity of the enzyme for the substrate, right? Now, Km has units of molar concentration that we have already talked about. Okay. Now, based on all of this, if you take the value of Km to be, if you take the value of V, that is the initial velocity, to be half of Vmax, and if you substitute that into the equation, then you get that Km is equal to S, substrate concentration. So that means when velocity of the reaction is half, the amount of substrate concentration is only your Km. Okay, that much is clear by now, hopefully. Okay, now we have questions for you. Now you start answering these questions. Okay, so the first question, people are leaving as soon as they're hearing of questions. Why? Questions are the fun part, right? Okay. The first question, so you can answer in the chat box or you can unmute yourself and you can talk. Okay, so see, the first question is question on enzyme kinetics. So enzyme velocity can diminish over time due to, firstly, decrease in substrate concentration, product inhibition, pH changes, and thermal inactivation. Right, so if you remember last class, then you will be able to answer these questions. Okay, because in the last class we talked about thermal inactivation, that there is a specific optimal zone of temperature and below that zone and above that zone the enzyme cannot function. Similar thing for pH. Product inhibition means too much of product has been formed and it is preventing the enzyme from making any more product. That's like a feedback mechanism, right? And decrease in substrate concentration. So which of these can diminish the velocity of the enzyme over time? Very simple, very straightforward question. You can try to answer. Yes. Let's see if we are getting any answers. Hopefully we'll get answers in the chat box. Okay. Chalo, it's a very simple question. Try answering. So which of these different things can affect the enzyme activity negatively? Okay, let's look at the answer. All of the above. Right, so all of these different things, as I told you right now, that all of these different things are going to negatively impact the enzyme activity because enzyme activity is restricted to very sensitive temperature, pH, substrate concentration and product inhibition, and all of these other factors as well. Okay, right. Now we will once again take a look at something that we have already discussed in the last class. This is kind of a revision for you. Okay, you remember, for temperature, enzyme activity was optimal only at a specific temperature range. Same thing for pH. And why is that so? Because temperature can denature the enzyme, right? So the number of vibrations should be optimal so that the enzyme and substrate molecules come and collide, and then the reaction is happening. If the temperature is too low, there will be less vibrations, less collisions, right? If the temperature is too high, enzyme gets denatured. It's after all a protein mostly, right? What about pH? The amino acid charges change. That is why the enzyme's conformation can change and it can undergo inactivation. Then the enzyme concentration. If you keep on increasing the enzyme concentration, initially it will increase, but later on, when the substrate concentration is after all fine, it's not changing, right? So what happens is all the enzyme sites get occupied by all the substrate molecule available, and so later what happens is, even when you increase the enzyme concentration, it will undergo saturation phase. Exactly same thing for substrate concentration as well, right? So I hope that explains the previous question to you. Now we will talk about this feedback inhibition. So I told you that nature has devised everything in such a way that there is no loss, no excess. Now we keep on wasting resources. We are humans, but the nature is designed in such a way that there is not much loss in the body. Similarly, let's say that glycolysis is happening. Okay, if too much of the product has already been formed, now the product means ATP. So let's say too much of ATP has been formed. Then what happens is, the ATP and the other products, they inhibit the enzymes from functioning so that excess of the products don't get generated. Right, so the presence of ATP at the end of the pathway inhibits phosphofructokinase enzyme, lowering the affinity for fructose 6 phosphate and therefore slowing down the glycolysis process. This is how product inhibition is making sure that excess of the products are not formed during an enzyme reaction. Now there is something called end product inhibition. What is it? It's exactly the same thing, right? This feedback inhibition is also called end product inhibition. Now it can be that, now another example is that end product inhibition is observed in humans, where the enzyme responsible for the production of cholesterol by liver is inhibited by the presence of cholesterol in the blood. Okay, so we don't need more cholesterol in the body because there is already so much cholesterol in the blood, right? So this is a very important mechanism to make sure that there is no excess of something in the body. And in feedback inhibition, the end product binds to the, so end product is the one that has gone through many stages, and it goes and binds to the enzyme allosterically. That changes the conformation of the enzyme, and therefore it cannot further form the products anymore. Okay, so that is end product inhibition for you. Okay. Now, there is something called feedback repression. Now, what is the difference between this feedback inhibition and feedback repression? Right. So, see, feedback inhibition is the mechanism in which the accumulated end product binds with the enzyme and inhibits the enzyme activity by binding with it. Right. On the other hand, in feedback repression is the mechanism in which the accumulated end product works as a repressor and inhibits the enzyme synthesis. So it is even working at the DNA level. It's acting as a repressor protein, and it is preventing the expression of the gene that folds into, that gives mRNA, and that finally gives the protein that forms your functional enzyme. Right. So one is acting at the protein level, one is acting at the DNA level. That's why it's repression of gene expression, right? And inhibits the enzyme synthesis at the genetic level. So this is the key difference between feedback inhibition and feedback repression. Clear? Okay. Moreover, end product binds with the active site in the enzyme in feedback inhibition, while end product binds with the DNA of that gene that encodes the enzyme and prevents the synthesis of the enzyme. Therefore, this is also a difference between feedback inhibition and feedback repression. Okay. Hopefully that is clear to you. Right. So now we have one more question. Okay. So deviation from the Michaelis-Menten kinetics occurs when? First, there are multiple substrate. Now, not all enzymes follow Michaelis-Menten equation. So we have talked about allostery, if you remember, right? So in allostery, you remember that some enzymes are regulated not by these just by Michaelis-Menten equation, but there are so many different other ways in which the enzyme activity can be regulated. Right? So allostery is one of them. Now see, when does Michaelis-Menten equation and the Michaelis-Menten laws not hold true? So there are different options. One is when there are many substrates, when the substrate binding is irreversible. K2 is equal to Kcat, where Kcat is your turnover number, and the enzyme displays cooperativity. So, which are the scenarios in which Michaelis-Menten laws will not hold true? So, you can answer in the chat box if you remember. C. Okay, let's see. Yes, that's very correct. C is the right answer. So, see that deviation happens when the substrate binding is okay, so only Michaelis-Menten equation has all these assumptions, right? That one is substrate binding is irreversible. Sorry, it's reversible. That is an assumption that we have. Second is single substrate is there in the reaction. That is also one more assumption. K2 is equal to Kcat. That is another assumption. So you can see we have already discussed so many assumptions, and here we are seeing even more assumptions. So for one theory to one hypothesis to be a theory, there has to be so many assumptions also to make it work at every scenario, right? So non-cooperative binding of the substrate is also one more thing that has to happen, because cooperative binding is seen in case of allostery, right? And allosteric enzymes do not follow Michaelis-Menten equation. Okay, now we will talk about the assumptions again. Okay, more assumptions of the Michaelis-Menten equation. So there is something called relative concentration of enzyme and substrate. Okay, no, this I think we have talked about. So the concentration of substrate is much more than that of the enzyme. That is one thing that we have talked about just in the 2-3 slides back, right? Then there is something called steady state assumption. So this is very important. So ES, concentration of enzyme substrate complex, does not change with time. That is your steady state assumption. That the rate of formation of the ES is equal to that of the breakdown of ES into E plus S and E plus P. Okay, so these two reactions are happening, and the sum of these two is equal to the sum of is equal to the rate of formation of the ES product, right? And in general, an intermediate in a series of reactions is said to be the steady state when its rate of synthesis is equal to the rate of degradation, right? Now, initial velocity. So, initial velocity uh, are used in the enzyme reactions, right? So this means that the rate of reaction is measured as soon as the enzyme and substrate are mixed. At that time, the concentration of the product is very small. So we had assumed that the concentration of the product is negligible, and we had assumed that the concentration of the substrate does not change over time, right? So the concentration of the substrate that we saw, that is important for finding out the velocity of the reaction, right? So the rate of the back reaction from the product to the substrate, that is also ignored. So some of these assumptions we had already discussed in the previous slides, but here is just once more given for your revision, right? Okay. Now we have another question. So the reaction is represented here as it is supposed to be, and the question is that the rate of ES breakdown will be E total. Okay, so they are asking you what is the rate of ES breakdown. Okay, and you have to give the equation for that. E total is the total enzyme, that means the enzyme present as free enzyme and enzyme substrate complex, right? So now based on this reaction, you tell me what will be, how will you represent the rate of ES breakdown. A, okay, let's see. Yes, that is very correct. So the breakdown of ES is actually K minus 1 into ES plus K2 into ES. Okay, you can solve it. You can write the reactions and the reaction constant, equilibration constant and everything, and you can derive from there. Okay, it's a derivation you can practice. Okay, now we will talk about competitive inhibitors, non-competitive inhibitors and uncompetitive inhibitors, right? Competitive inhibitor is very simple, straightforward. This is the enzyme, this is its active site. Substrate goes and binds at the active site. It's a reversible reaction. Inhibitor looks exactly like substrate or maybe a little bit different, but its binding site is similar. So inhibitor also goes and binds to the active site. This is also reversible. So when you add more of the substrate, then what happens? Even if the inhibitor is bound, it will be displaced by the substrate. Done. Second one is uncompetitive inhibitor. So what is happening? There is an inhibitor site. Inhibitor site is separate from that of the substrate. When inhibitor goes and binds, and substrate, whether it is bound or not, the entire enzyme substrate complex is going to be rendered non-active, inactive, right? So inhibitor binds to an allosteric site of the enzyme substrate complex. It only binds to the enzyme substrate complex, causing inactivation. Correct. Now, next one is our non-competitive inhibitor. So, competitive, uncompetitive. In uncompetitive, there is ES complex and the allosteric molecule goes and binds, and it binds at a site other than the active site, and it renders the entire complex inactive. Done. Okay. For the non-competitive inhibitor, this effector molecule, which is the inhibitor, it can either bind to the enzyme substrate complex or it can bind to free enzyme also. If it is bound to the free enzyme, then maybe substrate can bind, right? So see, the inhibitor binds to the enzyme or the enzyme substrate complex at an allosteric site. Catalysis is prevented due to the change in the enzyme conformation. So when it is bound, the substrate cannot bind to it anymore because the enzyme's conformation has changed, right? So that is why it is inactive. I mean, it is rendered, yeah, it's not going to happen anymore. So see, for uncompetitive inhibition, once again, the inhibitor binds to an allosteric site of the enzyme substrate complex, but not to the free enzyme. The inhibitor is not structurally similar to the substrate and does not compete for the active site. Okay, I hope that is clear. Okay, now these are the plot. Excuse me. So these are the different plots that you have to get very, very well versed with. Okay. So we have the Lineweaver Burk plot on the right, and we have this normal Michaelis Menten curve plot on the left, right? So Km is represented in both, Vmax is represented in both, and substrate concentration and velocity, etc. are there. Now for competitive inhibitor, see what is happening. So for competitive inhibitor, there is a change in the Km, right? Because it is affecting the affinity of the enzyme for the substrate. And there is also, but then there is no change in Vmax. Vmax is the same in the presence of competitive inhibitor, but Km is increased. Now these are things that you have to remember because there will be questions in which they will ask you all this, okay? For your non-competitive inhibition, Vmax is decreased in the presence of a non-competitive inhibitor, right? Because what is happening, the substrate is not able to bind to the enzyme because the enzyme has a different conformation, right? So its Vmax is obviously going to get decreased. And the Km is unchanged because the affinity for the enzyme of the substrate, it is not changing. So Km is unchanged for a non-competitive inhibitor. Right. So we go to a summary slide. Now summary slide is very important. Go through it. So in competitive inhibition, the inhibitor binds only to the free enzyme, not to the enzyme substrate complex. In uncompetitive, it binds only to the enzyme substrate complex. In mixed inhibition, now comes in another player in the game, right? Mixed inhibition. This involves inhibitor binding to both free enzyme and enzyme substrate complex with different binding constants. So that is Ki and alpha Ki. Ki means, what is this? This is the constant for inhibition. Okay. And lastly is non-competitive inhibition. So see, in mixed inhibition, what is happening? The enzyme inhibitor complex is there. To this, the substrate can bind and enzyme substrate inhibitor complex can form. So this is a new thing, right? And its rate is given as alpha Km, which is changed Km. So here Km is changing. Okay. Lastly, non-competitive inhibition is a special case of mixed inhibition where the substrate binding has no effect on the inhibitor binding. So alpha is 1. That means Km will be both, right? So both will be Km in this equation. Okay. Now, okay. So we have the plots in enzyme kinetics. This is very, very important. Michaelis-Menten curve and Lineweaver-Burk plot. Okay. So since Vmax is achieved at infinite substrate concentration, it is impossible to estimate Vmax and hence came from the hyperbolic curve, right? So hyperbolic plot means the Michaelis-Menten curve. It cannot accurately tell you anything about the actual Vmax, right? Now, see, because of this difficulty, the Michaelis-Menten equation was transformed into an equation for a straight line by Burk and Lineweaver. So the Lineweaver-Burk plot or double reciprocal plot is a graphical representation of the Lineweaver-Burk equation of the enzyme kinetics. Okay. The plots are given. You have to practice these plots. Now see, if these inhibition models, they are represented in the Michaelis-Menten curve, how are they going to look like? Your initial one is there, no inhibition. Then comes competitive inhibition. So what happens? Km is going to decrease, right? Km is going to decrease and Km is going to increase. Km is going to increase and Vmax is remaining unchanged, right? Then there is non-competitive inhibitor. So Km is unchanged but Vmax is decreasing. Inhibitor reduces enzyme activity but does not bind the active site. Okay, so this is how it is going to reduce, right? Now, what happens in Lineweaver-Burk plot? So in Lineweaver-Burk plot, if you show all these models, see what happens. This is no inhibitor. When you add inhibitor, competitive inhibitor, it changes this way. There is a shift, right? This way there is a shift. Km increases, Vmax is unaffected. Okay. Uncompetitive inhibitor. This is the one without inhibition, it just shifts. So both of these are reduced. Km is also reduced and Vmax is also reduced in uncompetitive inhibitor. Non-competitive inhibition or mixed inhibition. Km is unaffected but Vmax is reduced. So it is going this way. Okay, that is what is happening. So this is what you have to remember and you have to be able to replicate this in the question answers. Now there is something called Eadie-Hofstee plot. What is this? Now this Michaelis-Menten equation can be represented in different ways, right? So this is one of the representations where the different axes are V and V by S. Okay, and the slope that this gives you is minus Km. So the reaction rate is plotted as a function of ratio between reaction rate and the substrate concentration. This plot rearranges the Michaelis-Menten equation in this form. So this is Eadie-Hofstee. Okay. Now equations in uncompetitive inhibition, right? So Vi is equal to Vmax into S, Km into 1 plus I divided by Ki. This is what is represented as alpha. This alpha is actually equal to 1 plus I by Ki. Okay, where I is the concentration of the inhibitor and Ki is the dissociation constant for the enzyme inhibitor complex. Okay, so alpha is used to describe how the binding of an inhibitor affects the enzyme's interaction with its substrate. Okay, Ki is the dissociation constant for the enzyme inhibitor complex. It's given over here, the equilibrium constant for enzyme inhibitor complex, right? The inhibitor increases the apparent Km, but the maximum velocity remains unchanged. This is the case of competitive inhibition. That is why this is the equation. Okay. Now equations for non-competitive inhibition. So I know that right now as I'm talking, it is sounding like this is way too much to remember. But once you start practicing, it will become simpler. Okay, so as you practice, you will realize that there is some logic in all of this and you'll be able to understand this better. Okay, so for inhibitor binding to free enzyme, this is the equation. For inhibitor binding to enzyme substrate complex and also for uncompetitive inhibition, this is only your equation, right? So in pure non-competitive inhibition, the both Vmax decreases, but Km remains unchanged. Okay, you can go through this once again for better understanding. Now we will talk about Ki and Km. Okay, so the value of Ki is the dissociation constant describing the binding affinity between the inhibitor and the enzyme. While Km is the Michaelis constant in the Michaelis-Menten equation, which is used to describe the kinetics of substrate or enzyme binding, Ki is a thermodynamic parameter reporting the true affinity an inhibitor has for binding an enzyme. Okay, that is between enzyme and substrate, but Ki is for enzyme and inhibitor. Okay, affinity for inhibitor and enzyme. In contrast, Km is a kinetic parameter. This gives the substrate concentration at which half of the maximal velocity is attained. So that means Ki is a thermodynamic parameter and Km is actually talking about the velocity of the reaction. So it's a kinetic parameter. Okay. Now, Km is determined not only by the substrate's binding affinity but also how quickly the enzyme substrate complex is turned over into the product. In rapid equilibrium where substrate binding is fast comparing to the rate at which the enzyme substrate complex forms into the product. Okay. Km and Ki are identical. So when is Km and Ki identical? Only when the substrate binding is very fast comparing to the rate at which the enzyme substrate product forms the enzyme and product. Right? Whenever the assumption is not met, Km will be larger than Ki. So IC50, what is that? That means the concentration of the inhibitor at which 50% of the enzyme activity is inhibited. That is your IC50. So Ki is the inhibitor concentration at which 50% of the inhibition is observed. So IC50 is your Ki. Got it? Similarly, Km is concentration of the substrate at which half of the velocity is met. Okay. Right. Now we will move on to the questions. Okay. Try to answer this question. In a mixed inhibition, the enzyme inhibitor complex EI has a number 1 dash affinity for dash compared to that of dash. So higher affinity, lower affinity for enzyme or substrate compared to enzyme or substrate. So in EI, dash has dash affinity for dash. Fill in the dashes. Enzyme inhibitor complex has what affinity for what? So enzyme inhibitor substrate complex, that is supposed to be formed, right, in mixed inhibition. So that means we already know one of the dashes. What about the other dashes? Tell me. So whether the substrate is going to bind to the enzyme inhibitor complex with higher affinity or the enzyme with higher affinity. Right, so that is actually the question that is given here. So we can have a look at the answer. So see, we were thinking in the right direction. So 2 is substrate and the enzyme inhibitor complex is actually bind to the substrate more easily and with higher affinity than the free enzyme is going to bind to the substrate. Got it? That is why, of course, more of the substrate will be captured by this enzyme inhibition complex, which is anyway not going to react. And what is happening? More of the substrate is wasted, right? So in mixed inhibition is known as an inhibition in which the enzyme inhibitor complex has a lower affinity for the substrate compared to that of the enzyme. Okay, sorry, I just said the exact opposite thing, sorry. So enzyme inhibition complex has a lower affinity for the substrate compared to that of the enzyme. Okay, all right. Right, so now we go for the next question. So the next question is, in a competitive inhibition, an inhibitor binds to A, the enzyme at a site other than the active site, only to the substrate, reversibly at the active site, covalently to the ES complex. This you should be able to answer. This is very simple. This we have even discussed a few minutes back. Chalo, try to answer this. Okay, in the chat box. Chalo, try to answer this in the chat box. It's a very simple one. This every one of you will be able to answer. There is some kind of competition between the inhibitor and the substrate, right? So competition for what? You can write in the chat box. Okay, whatever option you think is the right option, you can type in the chat box. I'm waiting. It's a simple question. You should be able to answer this. Chalo. Chalo, chalo. Try to answer. This is simple. A. Okay. In competitive inhibition, actually what happens is the inhibitor and the substrate compete for the active site. Okay, not other than active site, not to any allosteric site. So the answer is C. Competitive inhibitor binds reversibly at the active site. Okay, so the right answer is C. Okay, now we have one more question. Which of the following equations represent the Lineweaver-Burk plot or double reciprocal plot shown? Okay, so the Lineweaver-Burk plot is given for you. Now, how are you going to tell me if 1 by V naught is equal to what? Okay. So, two of these are correct, and which two of these are correct? C. Okay, so C is 2 and 3. Okay, let's see. Okay, the right answer is actually B. So why is it B? Because B is 2 and 4. 2 and 4 are correct. So see, this is y is equal to mx plus c. That is your straight line equation, right? And c is this y axis. So that is 1 by Vmax. So this is the c. And y is equal to mx plus c. So m is km by Vmax. That is the slope. And x is 1 by s. So that means 2 is definitely correct. And for option number 4, if you rearrange this and if you take 1 by V naught minus 1 by Vmax is equal to this other thing, and if you rearrange it, you will come to the conclusion that this is also correct. So 2 and 4 are correct. Okay. Right. So now we will go for the next question. The graph below represents reaction rate versus substrate concentration plots for an uninhibited enzyme, that is orange. So Michaelis Menten, no inhibition. We have talked about this, don't forget, right? And the same enzyme in the presence of two different inhibitors. One is inhibitor A and the other one is inhibitor B. Okay. So in one of the cases, if you see for a curve A, there is no change in Vmax. If you note, there is no change in Vmax, but Km is increasing. For the next one, what is happening is Vmax is decreasing. And what is happening to Km? Km is constant. Or is it decreasing or increasing? What is happening? Okay. So you're saying that D is the right answer. Okay. So yes, curve A is definitely competitive. And see, in all the options, there is competitive only for option D. So your option D has to be the right answer, right? So that is how you decide. Yes. So option D is the right answer. For competitive inhibition, Vmax remains same, Km increases, and for non-competitive inhibition, what is happening is Vmax changes but Km remains same. So that is something that you have to remember, and this is how you will be able to answer the question. Okay. Now we have one more question. What is happening in this question? See, what kind of inhibition is depicted in this diagram? So there is an inhibitor which is binding to a substrate to a site other than the active site. So that means one of your options anyway get cancelled. Is it binding to the free enzyme? It's binding to a free enzyme. So that means another of your option gets cancelled. So that means two remain, right? Out of those two, which one do you think is the right answer? D. Okay, let's see. Okay, it's non-competitive inhibition. Okay, why is that so? Because let's go back to the plots. Yeah, so we go back to this summary figure. So in non-competitive inhibition, you see that this is what is happening in the non-competitive inhibition, right? So E plus S, and so see, in non-competitive, it's a special case of mixed inhibition where substrate binding has no effect on the inhibitor binding. So basically, EI plus S is also getting formed, and actually all the products are the same, but the only thing is that it is not affecting the inhibitor binding. Okay, so substrate binding is not affecting the inhibitor binding. Otherwise, it looks same. So that is a difficult one to answer. Okay, we'll move on to the next one. Okay, so see, this is the next question. In the scheme for the mode of inhibition shown below, where ESPI are the enzyme, product and inhibition respectively. Enzyme, substrate, product and inhibitor respectively. So the equations are given. Okay. Now see, they are asking you which of these is alpha Ks, alpha Ki, Ki and beta Kp. So if alpha and beta are coming in, that means it has something to do with the inhibitor, right? So which one do you think is going to be? So that is why 4, all of these are beta Kp, correct? So which is alpha Ki, which is normal Ki? What is happening here? C. Okay, let's see. Yes, C is the correct answer. So see, Ki is equal to Ei by Ei. Alpha Ki will come where? This 1 is your Ki, 2 is actually alpha Ki. This is your which one? C. C is alpha Ks and this is beta Kp. Right? So that is the C is the right answer in this case. Okay. Now we are going to talk about something simple. Right? Now we have talked about these Michaelis-Menten equations. We have talked about allostery. But then we know that Michaelis-Menten equation doesn't hold true when there is a multi-step reaction and when there is allostery, right? Besides other factors. Okay, so we have talked about allostery already. But then what happens in case of multi-step reaction? So see, multi-step reactions can be ordered sequential reactions, they can be random sequential reactions, and they can be ping pong or double displacement reactions. They can be majorly of three types. Right, so what is happening in ordered reactions? See, there is an enzyme, the substrate is binding, and there is another substrate that is binding, and then both of these are getting converted into the products and products are getting released. So that is ordered sequential. Next we have random sequential. So to the enzyme, first this substrate can also bind, first this substrate can also bind, and then both of them will be converted into products and they will be released. That is random sequential. Okay. Then comes in ping pong bye-bye. So this is the enzyme, substrate is binding, substrate is getting converted into product. Now the product is actually substrate for another enzyme and the product is binding to another enzyme, and what is happening? It is getting converted into another product. One second. Okay. Okay. So I'll be back within 2 minutes. Okay, you guys can wait here. I'll come back within 2 minutes. It's a 2-minute break. are back. Okay, we were talking about these ping pong bye-bye reactions, right? Okay, so this is what we have discussed about until now. We'll move on to the next one. Okay, so now see, we'll talk more in details about this particular reaction. So these reactions fall into two groups. One is sequential and the other one is ping pong. So these names indicate whether the reactants must bind to the enzyme at the same time for the reaction to occur, that is sequential, or if the product must leave before the second reactant comes and binds, that is ping pong. Okay. Now sequential reactions, in these reactions both the reactants must be on the enzyme for the reaction to occur. If this is a group transfer, a chemical group is getting transferred from one reactant to another reactant, the implication is that the group is transferred directly between the two reactants. These reactions are classified as ordered or random based on the binding of the substrates to the enzymes. Okay, so sequential mechanism, all substrates bind before chemical events. One is ordered. Enzymes bind in a different order with substrates. If the mechanism is ordered, the substrate will add to the enzyme A first, B second, and the first product to dissociate would be P followed by Q. Okay, the image is given in the previous slide. You can obviously refer to that. Now that is ordered reaction. One of the examples is NAD plus dependent dehydrogenases. Okay. Now if we go to the next one. So this is the one for ordered sequential reactions. Everything is highly ordered, right? Next one is sequential random bye-bye. What is happening? See, the order of binding is not important, right? Indicating that the binding sites for each substrate and product are fundamentally independent of each other. That is why it can be random. That is why randomness is allowed over here. Right, so sequential mechanism, no intermediate is formed. Okay, so one of the examples is this creatine to phosphocreatine, right? And this particular ATPase is involved over here, that is happening. Okay, now we have the next one. Next one is ping pong mechanism, ping pong bye-bye, double displacement. Okay, so like let's see what is happening in this bye-bye reaction. In the second major class of bi-substrate reactions, one of the products is released before the second can bind. A stable enzyme intermediate occurs in these reactions that reacts with the second substrate, right? So the enzyme is undergoing conformational change constantly. So often the enzyme is transiently covalently modified in order to transfer a functional group from one substrate to another substrate. Okay, so the functional group is carried from one substrate to another by the enzyme, and the enzyme is undergoing covalent modification for that. Note, the substrates are not in contact with each other on the enzyme. Okay, this type of mechanism is seen in serine proteases and common in aminotransferases and some flavoenzymes. Uridyl transferases, etc. All these are involved here in this displacement reaction. So see what is happening. This enzyme is there. The substrate A is coming in, and the enzyme A complex is getting formed, and the product is getting released as P. Enzyme has undergone some kind of modification. So the enzyme is now F and not E anymore, right? Now what happens? Next substrate comes and binds. Again enzyme undergoes modification. The substrate is converted into product. Product is released. Enzyme comes back to its original state, which is E. That is what is happening. Okay. Now there is a intermediate in this mechanism. Okay. Okay. So we look at these reactions once again. So in sequential, you know that the reactants A and B both bind before the product formations and then the products are released. Random sequential, random order of reactants. So first A can come or first B can come. Anyone can come. Okay. But ordered sequential means that specific order of the reactants have to come and bind. Only then the reaction will happen, otherwise it will not happen. Ping pong, the reactant A binds, followed by the release of the product, followed by the binding of reactant B, and then release of the product. What is the difference between this ordered sequential and ping pong? The difference is that in ping pong, the enzyme is undergoing modification. So between the two reactions, the enzyme is not the same. It is actually chemically modified, right? So that the next substrate can come and bind to it. So that is the difference. Okay. Now we will try to answer this question. Schemes of 3 different bi-substrate reactions are shown below. So see, 3 different schemes are given for you. So based on these 3 different schemes, which is what reaction that you have to tell me. It's very simple to understand. First thing is look at the enzyme, look at the arrows, and the third one is, that's it. Just 2 things you have to look at. Look at the enzyme, okay, how the enzyme is represented, and look at the arrows. Tell me which of these reactions is random, which is ordered, and which is ping pong bye-bye. Remember, in ping pong bye-bye, enzyme gets modified. In random, any one of the substrate can come and sit down first on the enzyme. But in ordered, everything has to be ordered. Try to answer this question. D. Okay, let's see. So, you're saying that one is ping pong reaction. That is correct. The second one is random. Yes. And the third one is ordered sequential. Right. Right. So, yes, that is 100% accurate. D is the right answer. In ping pong reaction, double displacement pathway is followed. In random sequential reaction, the substrate binds to the enzyme in a random fashion, where in ordered sequential reaction, the binding of one substrate to the enzyme becomes obligatorily prior to the other substrate. Right? So, yes, so you people have understood this. I am glad. Okay, we move on to the next topic. Right, now we will talk about some drugs as inhibitors. So in the beginning of the class, we saw that all of the common drugs that we know, a lot of the common drugs that we know, for example, your aspirin and then statins, and there were so many different drugs, right? So these drugs are something that we have heard of very often, right? So now we will talk about drugs that act as enzyme inhibitors in the aspect of COVID-19. Okay, so you know that for the virus to propagate, the virus first enters into the host. Host is the human body cell here, right? And then what happens? The virus genome is there, and the virus genome undergoes translation and formation of proteins, right? Transcription, translation, and formation of the protein. Now, cleavage of the polyprotein. So in virus, you know that the proteins, it is not like normal mRNA, only one mRNA codes for one protein, but in virus that does not happen. There is a long mRNA and there are protein for one, first protein, second protein, third protein, all of them are there in the same mRNA. That is called polyprotein, right? And what is happening? That is getting formed, and then the viral proteins are produced to cleave those, to ensure that the separate functional proteins can be formed, right? So to prevent that, there are enzymes, there are drugs like Ritonavir and lopinavir. These are the different drugs. Then for transcription and replication of the viral RNA, the virus requires RNA-dependent RNA polymerase. The SARS-CoV-2 requires. Why? Because it undergoes reverse transcription from RNA to DNA, right? And then normal transcription and translation happens. So nucleotide analog prodrug, which is remdesivir. A lot of you have heard of its name already, I'm sure. So remdesivir is one of the very famous drugs. Next, translation and production of structural and accessory proteins, viral assembly and release. This is basically the life cycle. In the life cycle, different drugs are targeted at different stage to ensure that does not happen. Now, why are we suddenly discussing about this? Because you have a question on this. Otherwise, why would we even discuss, right? So the question is dash inhibits HIV protease enzyme through a dash pathway by forming an enzyme inhibitor complex that prevents cleavage of a specific polyprotein. Okay. Dash inhibits acetylcholinesterase through a dash pathway by reacting with serine at its active site. So does this sound scary? Are you thinking if you have to remember all the names and all that? Try answering the question. A. Okay. So A is ritonavir inhibits the HIV protease enzyme through an irreversible pathway, and Y is diisopropyl fluorophosphate. This inhibits acetylcholinesterase through this particular pathway, which is reversible by reacting with serine at the active site. Okay, let's see. Yes, so B is the right answer. Why? Because ritonavir is a reversible inhibitor that inhibits HIV protease by forming an enzyme inhibitor complex that prevents cleavage of specific polyproteins, and diisopropyl fluorophosphate or nerve gas is an irreversible inhibitor and it reacts with serine at the active site of acetylcholinesterase to form a covalent bond. Okay, so sometimes for some enzymes remembering the mechanism is useful, because especially for this diisopropyl fluorophosphate, a lot of questions are asked from DIPF. Okay, so it's important that you remember. Okay, now we have the next one. The next one is another question. So which of the following statements about carbonic anhydrase is not true? 1. It can increase the rate of conversion of carbon dioxide to carbonic acid and bicarbonate ions up to 1,000,000 fold. It is present in red blood cell and plays a key role in the regulation of pH and fluid balance in different parts of the body. It is an active zinc containing coordinated by 4 histidine residues. And malfunction of this enzyme can lead to several diseases like loss of acid production in the stomach, kidney failure, etc. So the answer is shown in the figure. Okay, the answer for you is shown in the figure. If you look at the figure, you will see that the answer is already there. You can answer in the chat box. Okay, you can answer in the chat box. The answer is there on the slide. Shouldn't be difficult for you. Okay, so we'll look at the right answer. Since the answer is there on the slide, you can see that this zinc is actually forming this coordinate bond with 3 histidine residues and 1 water molecule. Right? So that means the answer is C. And in the active site of carbonic anhydrase, zinc is coordinated by 3 histidine residues. Okay. We'll move on to the next one. So isoenzymes and alloenzymes, we have almost come to the end of our class. So we have just maybe 5 or 10 minutes more and we'll be done with today's class. Right, so let's quickly finish. Now what are isoenzymes and alloenzymes? This is also an important question. So see, isoenzymes are different variants of the same enzyme. Okay. Isoenzymes are different variants of the same enzyme that have identical functions and are found in the same person. The definition includes enzyme variants produced by different genes and thus representing different loci, referred to as isoenzyme, as well as enzymes produced by different alleles of the same gene are described as alloenzymes, right? So, the enzyme variants that are produced by different genes present in the same loci, this is called isoenzymes, and as well as enzymes produced by different alleles of the same gene, that is called alloenzyme. One second. I hope the sensor will just turn on. Anyway, it's not turning on. So we don't have much light anyway. So see, gene B present in, it is giving a particular form of lysozyme, and this is found in muscles. Lysozyme C is found in stomach, lysozyme D is found in embryo, lysozyme E is found in cancer. So isozymes arise from gene duplication or alternative splicing of a single gene. Okay, that is why they are there in the same locus, but then they are different genes now. That is because of gene duplication, or it can be alternative splicing of the gene. Now, alloenzymes are variants of the enzymes found in different individuals of the same species, right? So, due to genetic variation arising from different alleles of the same gene within a population. Okay. Now, we have another question. Which of the following is not an isoenzyme? A. They differ in amino acid sequence but catalyze the same chemical reaction, which is not a property of an isoenzyme, right? So they differ in amino acid sequence but they catalyze the same chemical reaction. They have same Km values. They vary in chemical and physical properties. They are regulated differently. So which one of these do you think would be wrong in the case of, okay, so B. B says that they have the same Km value. Yes, that is right. So they have different Km values, right? So isoenzymes, they have different Km values. Okay. We'll go for the next slide. So now there is one more question, that is, when a product accumulates in a cell beyond an optimal amount, its production is decreased by inhibition of an enzyme involved in its synthesis. After the product has been utilized or broken down and its concentration is decreased, the inhibition is relaxed and the formation of the product resumes. This inhibition is a mechanism by which the concentration of certain cell constituents is limited. What type of inhibition is this? A. Mixed inhibition. B. Uncompetitive. C is competitive. And D is feedback inhibition. You know the answer. When we started the class, we were talking about this only. Right? So, you know this answer. Try to answer D. D is feedback inhibition. Correct. That's correct. Yes. So, in feedback inhibition, see what is happening. The enzyme activity is suppressed as it participates in a sequence of reactions in which the substance is synthesized and the product of that sequence. And this inhibition mechanism limits the concentration of certain cell constituents. Okay. Now we'll talk about regulation of enzymes in the body. We will not talk too much about this, but just something that is needed for your NPTEL questions, because these are all previous year's questions and also related questions, right, from other courses and all that. Okay, so M and L type of isozymes are found in muscle and liver. Pyruvate kinase. Pyruvate kinase, two types of isozymes are there. One is found in muscle, one is found in liver. The one found in muscle is called isozyme M. The one found in liver is called isozyme L. Right. So liver only, what is happening, see, it is actually this pKa is involved in converting this pyruvate kinase to pyruvate kinase L, right? And using ATP, this is being done. And in other glycolytic tissues, including liver, all glycolytic tissues including liver, this other reaction is happening where pyruvate kinase is used up for glycolysis and pyruvate is formed, and pyruvate is helping in transamination, formation of alanine, etc., etc. Right? So pyruvate kinase L is getting converted into pyruvate kinase L unphosphorylated, L or M, and that is being done by generation of a inorganic phosphate. So phosphorylated form is inactive, dephosphorylated form is active. Okay, and then reversion is actually using ATP. Now see what is happening. This pyruvate kinase M type, what is happening? It is like influenced by different kinds of allosteric modulators. So it is activated by AMP, that is your adenosyl monophosphate, fructose 1,6 bisphosphate. It is inhibited by the presence of ATP, acetylcholine A, and alanine. Why? Because these are all required for its reaction, right? These are all required as its product. Now, L type. What is L type doing? L type phosphorylation makes pyruvate kinase more susceptible to negative allosteric regulators. And what are the allosteric regulators here? Okay. Anyway, so the last enzyme in glycolysis is pyruvate kinase. It is irreversibly dephosphorylates phosphoenol pyruvate to form pyruvate, right? And several lysozymes encoded by different genes, for example, M, muscle and brain, and L is found in the liver. They are all allosterically regulated. So you saw that for M type and for L type, allosteric and hormonal regulation is happening. So allosteric regulation is by your phosphates, and hormonal regulation is by insulin and glucagon, which are the two hormones. Okay, cyclic AMP dependent pathway, this is happening. And hemolytic anemia, so pyruvate kinase deficiency can lead to hemolytic anemia. The red blood cells swell up, and then what happens? They break open, they lyse. So sodium potassium pump activity is inhibited, and it is very detrimental for the patient. Okay. Now see acetylcholine release and degradation in normal neuronal tissues. So what is happening? You know about synapse. In synapse, what is there? There is a, between the two nerve endings, information is exchanged through the form of electrical impulse, electrical and chemical. So synapses can be electrical transfer, there can be chemical transfer between synapses, right? So see these presynaptic neuron is there, and then there is a postsynaptic neuron, and the region between is called synapse. So what is happening? This acetylcholine, and it is getting released, and what happens is it makes sure that this acetylcholine is formed in the form of, it is surrounded by the synaptic vesicle. It is being carried to the membrane and it is being released, and as soon as it gets released, it binds to the acetylcholine receptor on the other neuron, and then a lot of downstream pathway happens, and then the signal is it's sent basically, right? And this choline, acetylcholine is converted into choline, right? Acetate group is released, and this choline again goes and helps in the formation of acetylcholine once again using acetyl coenzyme A, right? So that is what is happening. And active site of RNase, you see that there are two histidine residues and one lysine residue. RNases are very, very ubiquitous. That means they are present everywhere. Okay, that is why working with RNA is very difficult, because there are different forms of RNases, RNase L, RNase A. There are so many different types of RNases which actually cleave RNA, right? And these are even active when you heat them in an autoclave at a temperature of 120 degrees centigrade and around 15 lbs pressure. That is why double autoclaving of water is required. That means two times you autoclave the same water to get RNase free water. Okay, so we have the question: which among the following statements is or are true? Acetylcholinesterase hydrolyzes the neurotransmitter acetylcholine in the synapse and terminates its activity, and it acts as a non-competitive and irreversible inhibitor. Due to the pyruvate kinase deficiency, hemolytic anemia is observed. Both M and L type isozymes of pyruvate kinase are allosterically regulated. There are two lysine residues and one histidine in the active site of ribonuclease A. So which of these are true? Which of these are true? D. So D, you're saying that all of them are true. Let's see. It's A. All of them are not true, because we saw here that in RNA's active site there are two histidine and one lysine. Right? In the question they were saying there are two lysine residues. But then that is not the case. It is not two lysine residues, but it is one lysine residue and two histidine residue in the ribonuclease A. That is why your option number A is the right option. Okay, thank you. That's all for today's class. And we have all the sources slides. So you can refer to all those different sources to know from where I have collected all this information, all the slides, all the figures, all the writing, everything. Okay, you can go through those. And thank you for your time. This is the end of the class. If you have any questions, you can ask me, otherwise we can quit. Thank you so much for your time.
Thank you, ma'am.
Thank you. Thank you so much. Let's quit then.